speed of DNA existing as 46 chromatin strands. Normally, this DNA is dispersed in a somewhat loose configuration. However, when the cell is preparing to divide, each strand of DNA is condensed by a factor of 10,000 times into a structure called a chromosome. This folding and repackaging serves to facilitate the process of cell division. When the cell divides, the chromosomes separate, providing a complete copy of the generic information to each cell. The ribosomes are large, complex molecular machines found within all living cells. Their primary function is to synthesize new proteins by reading the instructions from a messenger RNA strand. Ribosomes do this by linking together amino acids in the specified sequence. Ribosomes consist of RNA and proteins. The smaller subunit reads the instructions from the messenger RNA, and the larger subunit assembles the amino acids into chains. When the ribosome completes the assembly of the new molecule, the two subunits of the ribosome split apart. Ribosomes are the only organelle that's present in all cells, including bacteria. The nucleolus is a dense, spherical, non-membrane-bound structure found in the nucleus of the cell. It's composed of primarily proteins and nucleic acids. Its main function is to trans-5 ribosomal RNA and combine it with proteins to form ribosomes. The nucleolus also can capture and immobilize proteins, thereby preventing cellular interaction with their binding partners. This helps regulate cellular activity. Mitochondria are the power plants of the cell. They convert sugar into ATP and enzymes related to cell metabolism. Mitochondria are unusual in that they are surrounded by two membranes and contain their own independent genome. They also can divide independently of the cell in which they reside, meaning that mitochondrial replication is not tied to cell division. The number of mitochondria found in a cell varies by the type of cell. The more energy a cell needs, the more mitochondria it will have. Many cells contain only one mitochondria, while others may contain as many as several thousand. Mitochondria are found in both plant and animal cells. The nuclear membrane consists of two lipid bilayers, the inner nuclear membrane and the outer nuclear membrane. The function of the nuclear membrane is to separate the nucleus and the nucleolus from the rest of the cell contents. The nuclear membrane is embedded with special proteins called nuclear pores, which act as specific doorways that permit substances such as nutrients, waste, and cell out of the nucleus. Vesicles are basically mobile storage units. They are similar to lysosomes in that they are constructed of a phospholipid bilayer, but differ in that while the lysosomes are involved in digestion and transport, the vesicles perform no modifications to their contents. Vesicles may transport or store many substances from complex organic molecules, like hormones, to simple substances, like excess water, until those substances are ready to be used by the cell or expelled. The lysosomes are tiny sac-like structures filled with enzymes called hydrolases. Like vesicles, lysosomes are constructed of a phospholipid bilayer similar to the cell membrane. The lysosomes function as the digestive and waste disposal organelles of the cell, and enable the cell to utilize nutrients. Lysosomes also break down foreign substances, such as invading microbes, thus freeing up their base components for reuse. The endoplasmic reticulum is a contiguous complex network of interconnected tubes and membrane sacs that extend through the cell's cytoplasm. This complex makes up roughly 1/2 of the membranous tissue of the cell. There are two types of endoplasmic reticulum, the rough and the smooth. The rough endoplasmic reticulum, or rough ER, gets its name from the fact that its surfaces are studded with ribosomes. The membrane of the rough ER is contiguous with the nuclear membrane as well as that of the smooth ER. The rough ER's primary functions are to manufacture and then fold proteins into their final useful configurations. The proteins it produces are then either secreted from the cell, transported to locations within the cell where they are needed, or sent on to the Golgi apparatus for further processing. The centrioles are a pair of structures on either side of the nucleus made up of 9 triplets of microtubules. The centrioles play a key role in the formation of the mitotic spindle during cell division, as well as helping determine the spatial arrangement and location of the nucleus within the cell. Eukaryotic cells differ from prokaryotic cells in that they contain a nucleus and other membrane-bound structures called organelles. Eukaryotic cells can be a plant, animal, or fungi. Many single-celled organisms like protozoa and all multicellular organisms are eukaryotes. Nuclear pores are hollow protein structures that penetrate the nuclear membrane and act as doorways between the nucleus and the cytoplasm. They allow small molecules and ions to freely pass into or out of the nucleus, but also may allow larger proteins to enter the nucleus from the cytoplasm if the proteins have special sequences, or tags, that indicate they belong in the nucleus. In the same way as proteins are allowed to enter, RNA transcribed in the nucleus and proteins destined to enter the cytoplasm have export sequences that tag them for release through the nuclear pores. The Golgi apparatus consists of stacks of folded membranes called cisternae. Each disc-like cisternae contains different enzymes which selectively modify the macromolecules that are passing through them.